Good. So we are now moving on to the next part of our event today, which was introduced already briefly by Thierry. We will have testimonies by many of our applicants, and Uli Köster will be now sharing those testimonies.
Okay, thanks, Thomas. And you have heard about the large diversity of projects which have been submitted, and we have selected 3 to give you a bit an overview of 3 different disciplines and 3 different tasks which are addressed with such isotopes. And so the first presentation will be given by Beatriz Salinas from the Hospital Gregorio Marañón in Madrid, Spain. And she has proposed a project which will use a new vector. I mean, you know, for radionuclide applications or theranostics applications, you need always a vector, an isotope, and maybe some imaging capability. And we will address the different aspects. And in the first one, it's an unusual vector. It's not a peptide or antibody, but it's exosomes. And since we were very interested to learn what are exosomes and what you can do with it, Beatriz will tell us more about. Beatriz, are you ready to share the screen?
Yeah, I think so. Can you hear me?
Yes, very well.
Okay, great. So let me share the screen. Okay. So can you see my screen now?
Yes, perfect.
Nice. Okay, so good morning, everyone. Thank you so much for this invitation, and also to give me the opportunity to show you today a little bit about our results in these kind of weird nanoparticles and how PRISMAP consortium can help us to move forward to new experiments that we really want to perform, but as I will mention later, we still are dealing with some issues, and especially in Spain. It's not only in my lab, it's around the country. So just a small summary about me. I'm the head of molecular probes lab and the biomedical imaging and instrumentation department group here at Gregorio Marañón Hospital. I'm also a lecturer at the biomedical engineering university here in Madrid, and also part of the core of a small animal imaging unit at the National Center of Cardiovascular Diseases. So just a small introduction about the group, because it's a little bit, you know, different to the standard labs that we usually see. We're part of the this biomedical imaging and instrumentation group is part of the University Carlos III. So most of us are also lecturers at the university. We are part of the hospital and in the experimental medicine and surgery department, actually. And we are part of this, you know, we are the core of the animal imaging facility at the National Center of Cardiovascular Diseases. So we are a little bit of mix of everything. Our department is a very multidisciplinary group. We have physicians, physicists, chemists, I'm a chemist, we have biologists, mathematicians. So it's a really nice mix of different backgrounds, which help us a lot to perform, you know, not only new experiments in the preclinical field, but also in the development of new imaging equipments and animal models. So it's a really nice mix of backgrounds here. And in the case of molecular probes lab, which is my lab, so we are basically ready chemists. So our main aim is to develop new imaging agents, especially for oncology and infection diseases. And as I will show you, so you know, based on those results that we are getting, we want to move forward to the new field of therapeutic. So so the main aim of this PRISMAP project is basically, as we mentioned before, is the development of new radiodiagnostics agents based on natural nanoparticles called exosomes, radioactively labeled with the novel therapeutic and diagnostic isotopes. Like, you know, at the beginning, our idea was terbium-161, but we were suggested to use, you know, thanks to this PRISMAP consortium, they suggested to use this terbium-155 and terbium-149. So Just to give you a small introduction, a background about these exosomes, because I know that's a new topic even for us. We work a lot in nanoparticles for imaging, and even in this field, it's a new kind of nanoparticle. But just a small summary, since the idea is to use these natural nanoparticles as a vector for these diagnostic radionuclides. Just let me give you some details about why nanoparticles and not other peptides or other antibodies, right? So one of the main advantages of nanoparticles in general is the layered surface to volume ratio that they have that allows us to play a lot with chemistry, radiochemistry, right? Incorporating new components in their structure. There is an arsenal of surface functionalization possibilities thanks to the different functional groups that we can attach to the surface of these nanoparticles, especially in the case of these exosomes, as I will tell you later, since they are natural nanoparticles and their surface is made by different kind of proteins and biological components. We have a large number of functional groups that allows us to play a lot with the surface. They have these capacities to carry large amounts of cargo. So at the end, higher amount of isotopes means higher yield of therapy and imaging. And in addition, in the application of nanoparticles in general, in the field of oncology, we have this passive targeting called EPR effect, enhanced permeability retention effect, that allows us, due to the fenestration of the vessels in this microenvironment in the tumor, allows us to include, or to allows us to target this tumor tissue with these nanoparticles in a passive way. So we can play with an active targeting, modifying the surface as a chemist, and also we can take advantage of this passive targeting of nanoparticles in general, which helps us to increase the biodistribution, the bioaccumulation of our nanoparticles, our vector in tumor tissue. So regarding nanoparticles in general and nanotechnology in imaging and therapy, so there are thousands of nanoparticles, but there's one specific kind of nanoparticles that is the gold standard or the ideal nanoparticle for this purpose. They are called liposomes. You may have heard about that. So one of the main advantages of these liposomes is their structures. Also, they are nanoparticles. They're in the range of below 200 nanometers, which is the ideal size for this EPR effect that I mentioned before. And then also they have this structure that has an hydrophobic layer in the surface and hydrophilic core. So that allows us to incorporate in the structure different kind of components. Like first, we can include hydrophobic drugs or hydrophobic components in the surface in this hydrophobic layer, but also we can incorporate hydrophilic drugs, isotopes, whatever we want to include in the core of these nanoparticles. But although there are thousands of publications in the use of these liposomes, for example, I just added here, like I was searching in PubMed yesterday, just looking for liposomes and preclinical application for diagnostic. We have around 13,000 publications in the use of liposomes for diagnosis. We also have around 22,000 publication in the field of liposomes in preclinical field for therapy. But although there is, there are a lot of, it is a promising tool for diagnosis, for therapy. When we move to the clinical trials or the clinical field, there are only a few of them, or more than 20 nanoparticle, 20 liposomes in the application of these liposomes in the clinical field. So there's a real problem in the translation of these liposomes to the clinic field, to humans. And main of these disadvantages is due to the synthetic pathway that we use to produce these nanoparticles. So since you need to use different chemicals that may be toxic for humans, at some step, maybe the purification is not the ideal one, but there are problems with the components in the structure of these liposomes, which limits, the translation or application of these liposomes. And here is where these exosomes appear, right? These exosomes are extracellular vesicles from endosomal origin. So cells produce these small vesicles. They have a nanometric size around between 50 and 150 nanometers. They have an active role in extracellular communication. And one of the key points, from a nanotechnological point of view, like from a chemist point of view is their structure. They present this lipid bilayer. So their structure is similar to these liposomes, which, as I mentioned before, are the gold standard in the development of new therapeutic and imaging nanoplatform. So they share the same structure, but they are produced naturally, right? So all the limitations that we observed before in the synthesis of these liposomes just disappeared, right? And also in addition, they have a real active biological function in cell-cell communication and disease progression, especially in oncology. They are related with many different pathways in the, not only in the production of the primary tumor. There is proof that these exosomes are like the first component, biologic component that decides where the metastasis is going to happen. They are part of, they form this, which is called the pre-metastatic niche. So they decide where the metastasis is going to appear, or it's going to be produced, right? So basically, these exosomes are natural nanoparticles with an active role. So we are combining a lot of advantages in just one specific kind of nanoparticles. And as I mentioned before, almost all the cells produce these exosomes, but we focus our research in one specific kind of exosomes. We produce these exosomes from milk, which I know it's a little bit weird, but it's quite convenient, you know, like, because they have a lot of advantages, right? Actually, they come from goat milk. So versus other synthetic nanoparticles like the liposome that I mentioned before, these exosomes are biocompatible, biodegradable. So they present, you know, they avoid all the limitation that the liposome may have in the transfer to the clinic. And if we compare to other kind of exosomes, you know, there are a lot of groups working on tumor exosomes, inflammatory cells exosomes, but if we use these goat milk exosomes as a nano platform, you know, at the end for our vector, they are economic, of course, no, it's milk. They are scalable, they are robust. There are other groups working in other kind of milk exosome called milk exosome breast milk exosomes, and they show how robust they are. So this is great for us since we can play from a chemical point of view, we can play a lot with their extraction. They do not present immunogenicity and cross-species, they present cross-species tolerance. There are a few papers in other kind of milk exosomes that prove that, and they're quite resistant. As I mentioned before, this is a key point for us as a chemist to play with the surface of this exosome to incorporate isotopes, other nanoparticles, other biomolecules, right? So we can use these exosomes as a drug delivery system, for example, right? We can incorporate drug in their structure for early detection. If we incorporate imaging agents in their structure, we can follow them, and as I will show you later, and we can detect the main processes where they are involved, right?
Beatrice,
yeah.
Your microphone is making some noise. Can you attach it somehow that it's not dangling around? Because
okay,
it's,
yeah. Maybe I can keep it like that.
Noise.
I think. Yeah. Sorry. I usually I'm alone in my office, but today everyone was here around. So yeah, as I was mentioned. So we are, you know, molecular probes lab. We develop imaging agents. So we thought like, okay, so since these exosomes are nanoparticles, right? And there is a huge field working on the labeling of nanoparticles in general for cancer detection. And also these exosomes have a role in tumor progression. If we label these exosomes, we can follow them. And since they naturally get accumulated in the tumor tissue, they have a role in this tumor tissue, we would be able to detect the tumor tissue, right? We just need to follow them just by imaging, right? So imaging was the key technology for that. So as I mentioned, I'm a radiochemist. So the first approach was based on the nuclear labeling, you know, radioactive labeling of these exosomes. We chose technetium because it was a really cheap isotope. That was the first work that we performed in the lab. We just started to set up the lab, so we didn't have many grants, right? So we moved to the cheapest isotope that we could find. Also, this metal was ideal for, you know, there were a lot of papers working on technetium labeling of liposomes. And since I mentioned before, since they are quite similar in structure to liposomes, was the ideal isotope for that. So we set up, we developed a very straightforward and easy methodology to label these exosomes. Is basically based on a passive incorporation of the technetium in the structure of this exosome. We could coordinate this technetium with the phosphonate groups inside of this membrane of the exosomes. We proved that this incorporation was a passive incorporation, not based on any specific transporter like NIS transporter that are involved in iodine, for example. We checked the stability of our labeling. It was really robust, right? We observed like a stability higher than 98% after 2 days. We confirmed that our technology did modify the physical properties of these exosomes, like size or morphology. And moving forward, we thought, like, how could we use this technique now that we have our isotope radiolabel? And so the idea was to evaluate how, since a lot of people are using this exosome as drug-delivery resistant, we use this nuclear technique to follow the administration route. So we were able to, I'm not going to go into detail, but we evaluated the different administration route usually used for the administration of exosomes as a drug-delivery resistant. And we, thanks to this nuclear approach, we were able to prove that depending on the administration route, the biodistribution was totally different. So that could affect to the treatment that you are applying, right? So we also perform other labelings with, since not all the labs have this nuclear equipment, we also perform optical labeling, fluorescent labeling of these exosomes. I'm not going to go into detail, just like to the approach would offer was a different methodology to the commercial kit that we usually use for the labeling of exosomes. Usually they use hydrophobic compounds that they just form a layer around the exosome, but this labeling is not stable. So we set up a new stable labeling to be able to follow, to track exosomes, to use these fluorescent exosomes in vivo, also to check pharmacokinetic properties, biodistribution, etc. So one of the main collaboration that we set up with other lab was to use these exosomes for the detection of tumor tissue, right? So we established a collaboration with the National Center of Oncology, Oncological Research, and we test many different kind of models with different kind of exosomes, right? And what we saw here is, let me see if I can show you the pointer. So here we have the signal from the exosomes, and here the signal for the cancer cells. So as you can see, there is a clear match between the uptake of our exosomes and the tumor tissue. So which support our hypothesis that these exosomes are going, after the administration, they are going to try to get accumulated in the tumor tissue. And what we are working now is like the combination of both modalities, because that allows us to evaluate in vivo, but also from a subcellular level to visualize the main populations that are involved in the uptake of these exosomes. So just from a chemical point of view, we set up both technologies, with both labelings in the structure of the exosomes without modifying their structure. We confirmed the uptake in different, because we wanted to evaluate if this uptake was the same in all the tumors or there was a different effect depending on the tumor cells. And what we observed was that clearly, for example, glioblastoma cell lines like U87 have a higher uptake of our exosomes in comparison to other cell lines. And also what we observed that, as we already knew, there was a huge uptake of this exosome in inflammatory cells, which also helps us, to evaluate this uptake in tumors, since there is a huge infiltration of inflammatory cells in the proliferation of the tumor. Then finally, we moved to the in vivo models. Here in this case was in a glioblastoma subcutaneous model. And as you can observe, there is an uptake in liver. This is, quite typical for nanoparticles. But we also observe here in the tumor tissue a very significant uptake of our exosomes. So our hypothesis is working. Our exosomes goes to the tumor and they get accumulated for that. So they could help us to not only that, detect the tumor tissue, but also if we incorporate any kind of therapeutic component in their structure, we could use them as a drug delivery system. And in this case, instead of drug, you know, as you can imagine, we were thinking about isotopes, therapeutic isotopes. We also evaluated, you know, since those exosomes were dual, we could evaluate the pharmacokinetic profile by imaging, but also, as I mentioned before, the subcellular implications. And what we observed was that the main uptake of these exosomes based on confocal imaging was in tumor cells. We also observed a huge uptake, you know, a very significant uptake in macrophages, which was part of our hypothesis also, since we knew that exosomes from milk are quite involved in inflammatory processes. And we also observed some uptake in fibroblasts. So what we are doing now, you know, I showed you that there was also a big uptake in liver. So what we are trying now is to improve the uptake in the tumor using this vector, this exosome, these nanoparticles, to increase the uptake of this exosome in the tumor and reduce the uptake in the liver. So we are doing what it's called the exosome engineering, exosomal engineer. We started with a very simple molecule, but now we are moving forward to more exotic chemistry, let's say like that, like chemistry or the, you know, of different approaches. But I just want to show you here like a brief introduction of this methodology. What we have done is just like to enrich the surface of these exosomes with a biomolecule that is specific also for tumor progression, like the folic acid, which is expressed in there are folate receptors in macrophages, but also in tumor cells. So we set up all the chemistry around that. We proved the presence of this folic acid in the surface of these exosomes. And when we moved to the in vitro and in vivo assays, we proved that there was an active incorporation of this exosome, not only by this natural biological role, but also now we added another role based on this enrichment with this folic acid. And finally, when we moved to the in vivo validation, in this case with the HeLa model, since these HeLas overexpress these folate receptors, we observe a really nice uptake in the tumor. But most importantly, when we compare to the previous study that I showed you before, the ratio between the liver and the tumor decreased almost threefold, which means that we are increasing the uptake on the tumor and reducing the uptake of the exosomes in the liver, which will help to reduce the secondary effects maybe in other healthy tissues. So now we are working in this topic, in this exosomal engineering, to improve. Now that we know that basically these exosomes reach the tumor, actively they reach the tumor because they active role in tumor progression. But now, you know, also I know that we can help to increase the tumor uptake with this exosomal engineer. We are improving our studies, our tumor oncology studies, in the uptake of these exosomes. So just the take-home message, you know, like thanks to this labeling of exosomes, we can confirm not only the diagnosis capacity of these nanoparticles in tumor models, but also the evaluation of this biological behavior for further use as drug delivery system or imaging agents. And now here is where PRISMAP project comes, you know, helps us in a new research line that we want to perform. Is as I mentioned before, the aim of this PRISMAP project was to develop new radiotheragnostic agents based on these natural nanoparticles, just based on the radiolabeling of these exosomes, you know, incorporating therapeutic and imaging nucleides, radionuclides in their structure, since now we know that these exosomes reach the tumor tissue. So the steps in these PRISMAP projects would be the first step, of course, you know, set up the labeling of these exosomes. We have been working already, as I'll tell you later, with TUM University in Munich, and the labeling of these exosomes with lutetium. So our approach would be two different approaches: either labeling these exosomes in the surface using a chelator, right, like we are doing with the lutetium, and we would try also the incorporation of this terbium isotope in the surface, in the hydrophobic surface of this exosome, similar to the technetium approach that I showed you before. Once we, you know, set up this radiolabeling, we would move to the in vitro assays to prove that, to select the ideal cell lines, to evaluate the effect of these radiolabeled exosomes as a therapeutic theragnostic agent. Initially as a therapeutic, but ideally we would like to combine both. And finally, we would move to the in vivo model, selecting as I showed you before, the best model. So far, we know, based on our previous studies, we have seen that melanoma, glioblastoma, and HeLa models are the ideal one for the uptake of these exosomes. But we will move to the in vivo studies, evaluating not only, first by imaging, since these isotopes like the 149 and 155 allow us to use SPECT and PET imaging depending on the isotope. We could, first visualize if this treatment reached the tumor and then follow the successful of this treatment based on this radiotherapy, based on these nanoparticles, right? They would act as a Trojan horse to bring the isotope, the therapy, to the tumor. And regarding PRISMAs, how did PRISMA get into our life? So it was because we heard about PRISMA thanks to Dr. Weber and Dr. Cosas from TUMS, from Technical University of Munich. Because we were already performing some studies with exosomes and lutetium 177. So Dr. Weber suggested to contact, to apply for this PRISMA project and test not only lutetium, but also other interesting isotopes for therapy, and maybe the combination of therapy and imaging. And why is this PRISMA consortium so helpful for us, right? So as I mentioned before, we have a lot of limitations here in Spain for radiochemistry and for nuclear imaging and nuclear medicine in general, because we have limited access to cyclotrons, especially for new radionuclides. For me, it's quite tricky to work in this field because we can only work with fluorine 18 and gallium, because we bought a generator technician. And that's all, from the cyclotrons from here, from Spain. As you can see, technetium and gallium are coming from generators. So actually, we only can work with fluorine. We buy also zirconium 89 from other countries, but we are quite limited. So, PRISMAP would be the ideal platform to allow us to work with other isotopes that we don't have access here in Spain. Also, it's a new field of research here in Spain. I got my degree in radiochemistry and my training in the United States and in Germany. So what I'm trying now to do is to create like a network or like some kind of training program here in Spain to increase the interest in this field of radiochemistry and nuclear medicine and new probes here in Spain. And here I think also PRISMA is ideal platform for that. PRISMA is ideal platform to the development of this new research line, also to, as you mentioned before, for the training of the students, the PhD students, but not only PhD students, postdocs, because the only way to train someone here in Spain is the 2 or 3 radiochemists that are in Spain, so there's not an official training for that. So I think that PRISMA is also an ideal platform for, to create a new generation of radiochemists and or radiophysicists in Spain. So I think it's a great opportunity for us and a really useful tool for us. And just to finish, I would like to thanks to our lab, of course, especially for this PRISMA project. The team will be me, Maria Isabel Gonzalez, which is a PhD student who has been working, in all the experiments that I showed you before in an exosome radiolabeling, also the radiolabeling in Munich with lutetium. Professor Manuel Desca, who is the chief of the department, and also in collaboration with Dr. Wolfgang Weber and Susanne Kosatz from the Technical University of Munich. So that's all. Thanks a lot again for this invitation, and thanks for allowing me to be part of this PRISMA consortium.
Okay, so thanks a lot to Beatrice. I fear that if this takes off, we all have to curtail our consumption of the tasty Spanish goat milk cheeses because you have a new application of those exosomes. So do we have any questions in the audience here or in the chat? No questions now. You mentioned the training program. So I remember that Thomas proposed that local PhD schools can sign up to the charter of the PRISMAP PhD program, and that might help you to get a better mixture of your local PhD students into the European community of PRISMAP PhD students.
I think it's a
So
great
please feel
opportunity.
free to contact Thomas. Yeah.
Yeah, I will be more than happy because I think it's a great opportunity for them. So far, I'm the only one who can offer them like the training. So and my knowledge is limited. So I think it's a great opportunity for them also to learn new topics.
And another point is that we have no yet Spanish responders to our survey, which everybody can find on the first page of the PRISMAP website on the right. So please fill in. Like this, we can better plan what services to develop. And you mentioned that it's very much limited in Spain. So it would help us a lot to get feedback. What will be the demand from Spain?
I
now I saw that David has a question. Please,
Yeah,
David,
hello, thank you, Uli. Thank you very much for this nice presentation. I'm discovering the exosome and I find it very cool. So I have a question because you are speaking about cell uptakes. Could you clarify actually that the exosome is going to fuse with the tumor cells? It's going to stick somehow to the tumors, to the membrane of the tumor cells, or it's just going to stay around the tumor microenvironment? What is the, when you say uptake, what does it mean?
So what we have seen, let me share again, and I think I have a picture of that actually. What we have seen here, like in this case, for example, in this HeLa cells, can you see the picture? I don't know if
Yes,
so
yes,
what you can see here, what we have observed in the different cell lines is the perinuclear accumulation of these exosomes actually. So they are in the cytoplasm, and after a few hours, usually 24 hours in vitro assays, we can clearly see the perinuclear accumulation. So that's the reason that we are trying to move to alpha emitters to see if that would be effective for this kind of cells.
Yeah, it's excellent then, because you don't have to worry too much about the release of the radioactive radionuclides. Okay, that's yeah.
Thank you for the question.
Okay, so I don't see other questions.
Sorry.
Yes.
I have a question.
Joe, yeah.
Beatrice, very nice presentation. I liked a lot. Did you label your exosomes with pertechnetate? What technetium molecule did you use to label the exosomes?
So as I mentioned before, it was a very straightforward methodology. It was just based, we use this pertechnetate, we reduce it with thin chloride, and just based on this technetium 4, we could incorporate it. Yeah, exactly. So we just needed to reduce the technetium 7 to technetium 4, and then with this reduced technetium, we incorporated with the coordination with the phosphate groups in the membrane.
Okay. So, but you are not sure about the structure of the molecule?
Not really, not really. So the test that I showed you before, when I said like we knew that it was a passive targeting and not an active targeting, it was before, because we knew that some studies are, like there are a lot of studies using pertechnetate, for the accumulation by these NIS transporters. So we check if the incorporation, and since these exosomes came from the cell, we wanted to see if this incorporation of the technetium was based on the NIS transporter, this iodine transporter, or just a passive coordination in the membrane. And what we observed was there was no, let me, there was not accumulation of these not accumulation using the this is the study.
At the same time, I'm not listening very well.
So I think they are a bit pressed in time, so
okay, sorry, yeah, sure.
I think we have to go on. Thanks a lot.
Thank you.
And I'll let you discuss over coffee break.
Yeah, thank you. Thanks a lot.
So thanks again to Beatrice. And after having heard about this quite unusual vector, we will hear now from Enzo Torino, who is professor of chemistry in Torino in Italy, about chelator, which is used to attach radiometals to the vector. And since Enzo happens to be member of a Euroimaging consortium, he will tell us also a bit about this longer standing collaboration, which can give us ideas how to organize things within PRISMAP. So it will be about chemistry and also a bit about how to manage European user access. Please, Enzo.
Okay, thank you very much. Can you hear me?
Perfectly.
Can you hear me?
Yes,
yes,
loud and clear.
Okay, okay, thank you very much. And also you can see my screen.
Also, yes.
Okay, thank you. So thank you very much, of course, for the introduction and for the invitation to this public event in PRISMAP Consortium. So first of all, I would like to very briefly introduce myself. My name is Enzo Torino, and I'm a professor of general and organic chemistry at the Department of Molecular Biotechnology and Health Sciences at the University of Torino in Italy, and my main research interests are in the field of the design and the preclinical validation of imaging probes, with a special focus on MRI and application of MRI in molecular imaging. I have also the pleasure and the responsibility to coordinate the multimodal molecular imaging Italian node within the Eurobioimaging infrastructure. And I would like also to briefly introduce to you who are not familiar with this consortium, Eurobioimaging. Essentially, this is a European consortium, a European research infrastructure consortium, I think somehow similar to PRISMAP. And the main mission is to provide services and open user access to any user, any researchers interested to work and to do research in the field of biomedical imaging. And essentially, Eurobioimaging is a consortium that is currently composed of 17 countries, essentially 16 countries plus MBL in Heidelberg. Of course, the organization is a typical hub and node organization. Essentially, there are two main communities in the consortium. One is the biological imaging communities, so expertise essentially in the field of microscopy, so optical microscopy, electron microscopy, super resolution, and so on. That is coordinated by MBL. Another family is the family called medical imaging, so people with expertise in essentially in vivo imaging at the preclinical and also clinical level. And this hub is coordinated in Italy, and the headquarters is here in Torino. And then there is, of course, a directorate hub that is located in Turku in Finland. So essentially, this is the organization, and of course, essentially the integration between in vitro imaging and in vivo imaging is extremely important, because it allows us to offer a very wide offer in terms of imaging services, with the ambition essentially to see molecules from molecular level to, of course, a very complex system like, of course, animals and, of course, humans. So this is essentially the portfolio, the technological portfolio of the consortium. At the moment, essentially, we can offer consolidated imaging technologies, but essentially this portfolio is continuously extending to other new technologies, like for instance, photoacoustic imaging or EPR imaging, or also mass spectrometry imaging. Each country has his own organization. Essentially, there are almost 150 research facilities in the consortium. In Italy, there are 4 nodes. One is on advanced light microscopy, so the biological in vitro imaging community. Other 2 are single site node, one with expertise in Trieste in Electra with synchrotron light, essentially with the phase contrast imaging, and another specifically devoted to neuroimaging close to Naples. The multimodal molecular imaging node is composed of 8 research centers, 1 in Torino, this coordinator, 2 in Milan, 3 in Pisa, and 2 in Naples. And we, of course, belong to the family of the medical imaging app. In Torino, essentially, our activity is distributed in 3 different research centers. The Center for Molecular Imaging in downtown, the Center for Preclinical Imaging that is located more or less 50 kilometers north from Torino. And very recently, we have another research facilities, again, very close to the Center for Molecular Imaging in Torino. It's called Center for Translational Imaging. And this was inaugurated a few weeks ago, and this is my current workplace, and I'm talking from one of the offices on the second floor of this building. And you can see here that essentially our technological portfolio is essentially can cover several imaging technologies, in vivo imaging technologies, essentially mostly at preclinical level, with a very good equipment, especially in MRI scanner with different scale, with different magnetic field. We are also got a grant in order to buy clinical 3 Tesla MRI that will be installed in this new building. And in this new building, we have already a preclinical 7 Tesla MRI, and what it is, of course, relevant for the PRISMAP connection and PET-SPECT-CT preclinical scanner. The expertise, our expertise essentially is in the field of designing imaging probes, mostly for MRI, so for small molecules, paramagnetic complexes, that is the golden standard for the clinical approved contrast agents in MRI. Alternative to metal-based agents like CEST agents, fluorine-based agents, hyperpolarized agents. Our main application field is in oncology. Of course, it's not exclusively oncology, but mainly in oncology, metabolic imaging, imaging tumor microenvironment, and there are also several research lines devoted to the support of imaging to therapy. Different kind of therapies, BNCT, surgery, very interesting, very important, and the classical support to pharmacological therapies. And we have also some experience in the last decade in nanosystems and in developing nanoprobes. So what about our experience in nuclear medicine? Essentially, our experience is at the moment limited to support research mainly in the site, in the Center for Preclinical Imaging, because in that site there is also a production and research site of AAA, Advanced Accelerators Applications, now AAA Novartis. So we have some experience in giving them support for the animal models and for the imaging side, but we have no experience, direct experience on the radiochemistry. And so now in this building, of course, we have a new instrumentation. We are in the phase of getting the authorization for starting the research, and we are really beginners in the field of radiochemistry. And so we established some connection, especially one successful connection was with the Technical University of Munich, with Dr. Calogero d'Alessandria. And we started a research collaboration. There is one PhD student of our group that is working there in studying essentially and labeling this ligand that you can see in this slide with gallium-68. So of course this connection was extremely important for us, for a sort of training, but also in order to know about PRISMAP. And for this reason, essentially, we decided to apply for a project. And the project was essentially the preclinical validation of this ligand that we synthesized last year with a main objective to labeling with gallium. But of course, in the context of the PRISMAP call, we decided to write a proposal in order to validate this ligand using this quite original couple of theranostic pair with the diagnostic terbium-152 and the therapeutic alpha emitter terbium-149. So this was the original plan, and essentially the application was in the field of FAP imaging. FAP is a quite emerging marker, quite ubiquitous marker, but very common in oncology. And we started and decided to use this vector. This was already validated and used in humans, but using DOTA as a chelator. And in our idea, we conjugate this FAP inhibitor with an ASTA chelator. And essentially, in this talk, I would like to describe some properties of this chelator. This is the synthesis. I don't want to enter into detail of the synthetic steps, but only to give you an overview about the synthesis of this ligand that we accomplished last year. There are many steps. This means at the end that we have an overall yield that is not so good. Of course, there is room for improvement, but fortunately and differently from MRI, sensitivity is not an issue in nuclear medicine. And of course, in our first batch was enough in order to start the collaboration with Munich for the labeling with gallium. And so this is okay for us. It's a very good starting point. Why we decided to use ASTA? The first paper published on ASTA was in 2004 and was a collaborative research effort from two universities, mainly the University of Eastern Piedmont. Giovanni Giovenzana is essentially the father of this ligand in collaboration with our group in Torino and with the support also from Bracco Imaging. So ASTA is a diazepinic ligand with a cyclic structure, but with the presence of 7 donor atoms. This is very interesting in terms of the design of MRI contrast agents, because the gadolinium, that is the golden standard for the paramagnetic metal center, has typically a coordination number of 9. And this means that in this complex, the metal center has the possibility to coordinate 2 water molecules in fast exchange with the bulk water. And this is extremely important for designing a high efficient MRI contrast agent, because the contrast is mainly generated by the number of the water molecules that visited the paramagnetic metal center. And this means that in this compound, the relaxivity, that is a measure of the ability of the compound to generate MRI contrast, is about 50% higher than the classical and clinical approved compound like gadolinium DTPA or gadolinium DOTA. Then another important advantage of this system is that, differently from other coordinatively unsaturated ligand with 7 donor atoms, this complex is not able to interact with biological anions like carbonate or phosphate. And this is very good, of course, because this means that it is quite inert in the biological environment. And it is important because the interaction with the biological anions causes a dramatic drop down of the relaxivity, because of course the anions can replace the water molecule coordinated to the metal. So this means that this kind of interaction is detrimental for the relaxivity and also for the biological interaction. This was the first paper. Since then, at the moment, there are almost 60 papers on ASTA. And now I will show you some selected examples to give you an idea about the properties of this ligand and the metal complex of this ligand. This is a paper in 2009 in collaboration with a group at the University of Debrecen, who mostly contributed in the development of this ligand, Zoltan-Barenai and Erno Brucker in particular. And essentially, they measured the thermodynamic stability of a series of metals, the valent transition metal, or mostly lanthanide, thermodynamic stability, and also kinetic stability and kinetic of transmetallation. That is, of course, an important point, especially for the MRI contrast agent. And you can see that essentially this ligand has a very nice property, the very high thermodynamic stability, slightly lower than DTPA and DOTA, but essentially very, very stable, but is extremely stable and in terms of kinetic stability towards the transmetallation, and especially if compared with the linear DTPA ligand. So this is an extremely strong point of this kind of ligand. Then, of course, people started to work with this kind of compound and try to improve several properties, and remaining in the MRI field, one improvement was essentially to design multimeric systems, because in this kind of system, there is also the advantage to design molecules with a slow tumbling motion, and in generating the contrast in addition to the number of water molecule coordinated to the metal center, it is also important the molecular orientation of the systems. If you slow down the molecular orientation, you have an advantage in terms of contrast generation. And you see here a couple of examples of systems based on ASTA, a dimer and a multimer, in which essentially the relaxivity was improved significantly. But we have to wait until 2012 to have the first paper in which ASTA was applied in nuclear medicine, and was essentially a paper from Bracco Imaging. And they optimized the structure of the cyclic RGD, the tripeptide that is very known to interact with alpha V beta 3 integrins, essentially for imaging neoangiogenesis. And they conjugated this optimized peptide with ASTA and label the ligand with gallium. And you can see here the first essentially preclinical experiments on a mouse model on a tumor expressing the corresponding integrin, and to see that essentially the targeting tracer was able to accumulate in the subcutaneous tumor with essentially a dynamic behavior that was quite different with respect to the control that was the ligand without the targeting vector. So this was the first experiment. Since then, a series of other papers appears in the literature. Also other groups, and especially the group of Frank Roche in Mainz, started to use this ligand. The beginning still with gallium, essentially, with other some also smart application. For instance, the possibility to design an hybrid system containing gallium and gadolinium, using gallium as a quantitative probe in order to have an information about the amount of the system, and to use a gadolinium agent as a smart probe to measure some physicochemical variables in the microenvironment, for instance, in this case, pH. This is quite an interesting approach. Other groups modified the structure, introducing a vector for targeting specific marker, for instance, in this case, for imaging bones. Or the group of Frank Roche also developed some derivatives in order to have a system that are ready for bioconjugation, especially this data 5 compound. And then also there was an extension from gallium to other radiometals, of course, and mainly scandium-44 for several papers on these radiometals, and also, of course, lutetium, that is a lanthanide, so it is extremely a good metal for this kind of ligand. And also the extension in the application to PSMA, that is, of course, a very important biomarker. In this paper, there is also one example in the using of ASTA for FAP imaging, that is also our interest, again from the group in Mainz, but essentially at the moment they only published the radiolabeling and some in vitro data on this compound, characterized by the presence of this peculiar squaramide linker between the FAP inhibitor and the ligand. And you can see here the real advantage in using ASTA, that is of course in the rapidity and in the radiolabeling. You have here the comparison between the use of an ASTA ligand, the compound that you can see here, and compare with the same system containing the DOTA chelator in terms of radiolabeling. And you have at the room temperature a sort of complete labeling in very, very short time, using also a small amount of precursor and also a good stability of the system. Whereas in the case of DOTA, of course, you require to heat the system, and the maximum the complexation was completed in much longer time. So this is essentially the main advantage in ASTA, in which essentially you are in the middle between a linear and the classical macrocyclic environment, but the kinetic inertness is not so high as in case of DOTA. So you can label very fast, but with a very high stability and high thermodynamic stability and kinetic stability. So return back to our contact with PRISMAP. Essentially, our idea was to propose this dual radiolabeling for diagnostic PET dosimetry and radiotherapy approach with this kind of ligand. We are very happy to know that the panel evaluated quite well our project, but essentially there were some technical problems, especially for the delivery of the 149 terbium at the biomedical site in the Technology University of Munich. So for this reason, essentially, the project at this stage essentially was limited to the optimization of the radiolabeling of the ligand with terbium 152. Then, of course, the typical assessment of the in vitro stability and binding and uptake to selected cell lines overexpressing the FAP biomarker, and followed by the preclinical validation in vivo on a specific mouse model expressing, of course, FAP. And essentially, we expect to complete this project approximately in 10 months, with an expected starting date in fall, in September or October. And of course, our desired partners for the research was the TUM and as a reference, Calogero, of course, D'Alessandri. Okay, we'll conclude here. This is a group, a photo group of our team in Torino. We like very much this picture because we took it in a break between 2 consecutive lockdowns. So everyone was there. It was a very nice day. And I would like also to take this opportunity to spend no more than 1 minute to advertise on an educational initiative that we promoted in our university in Torino, that I think that could be also of interest in PRISMAP consortium. Essentially, that is related to a master degree, classical 2 years master degree that will be fully offered in English. And the topic is essentially the biotechnology and the chemistry in diagnostics. Also, of course, biotechnology, chemistry, and biology in diagnostics. And essentially, this is a quite original educational offer, at least for sure in Italy, but there is also a few examples in Europe. And the idea is to provide knowledge on diagnostics, in vitro and in vivo diagnostics, from the perspective of a chemist, biologist, and biotechnologist, and less from the perspective of the clinicians of the medical side. It is essentially much more common. And of course, nuclear medicine, imaging technologies, and radiochemistry will be an important offer in topic in the educational program. And for those of you who are interested to get more information, you can visit this website. So thank you very much for the attention, for the invitation.
Okay, so thanks a lot, Enzo. And I'm sure that especially the last slide will find the interest of Thomas. Do we have a short questions? And else we have to go on, I fear, to the next talk. Anybody has a short question?
Yeah, just a very short question, because it's very interesting, this last time. I'm wondering why I'm not using more. Does it is it commercially available, or it's still something we need to produce in a chemistry lab?
No, there is essentially the Giovanni Giovenzana, so what I call the father of ASTA, as a spin-off activity, and essentially, of course, is very available to support the research and to sell, of course, the ligand. So it's commercially available. Not only the ligand, but also a series of derivatives in order to bioconjugate the ligand with any kind of classical targeting moieties or any kind of chemical moieties.
Thank you.
The spin-off is called the Cage Chemicals, is the name of.
Okay, so thanks a lot. And after having seen the importance of a vector and the importance of a chelator, now you have your complete radiopharmaceutical. You inject it somewhere, but you still want to know where it goes. And this will be addressed by our third talk, which will be given by Michel Kohle, who is a medical physicist at KU Leuven in Belgium. And he speaks about how to image, in particular, alpha emitters, which is a particular challenge. Michel, you are ready?
Yes, I think so. Yeah, I will share my screen also. 1 moment. Okay, can everybody see my screen? Or
Yes, we see your screen.
okay, great. Good morning, everybody. I will give a talk on the imaging of alpha emitters, and mainly focus on actinium-225, because that is the main research focus at our department for the moment. So perhaps a short introduction on targeted alpha therapy. So the use of alpha particles has definitely some advantages. First of all, they have a very short range within tissue, so you can really localize your dose. They also have a high lead, and so the damage to DNA, is very effective by these double-stranded breaks. And so this is, I wouldn't call it a famous, but a picture which is often shown also to demonstrate that they can also be considered as adjuvant therapy to overcome radio resistance to treatments using beta emitters. That is basically a patient where there's still disease present after 4 cycles of lutetium, after treatment with lutetium dotatate. And basically the disease then disappears after 2 cycles of actinium labeled dotatate. But they are very effective, but of course they are very toxic also. So that basically means also that we need to monitor closely the biological clearance. We also have to take into account the affinity for different organs. And what is also of interest for alpha emitters is the recall daughter effect, because if you get the emission of an alpha particle, you will also get some energy, which is sufficient actually to break the binding between the radionuclide and the targeting agent, as is shown here. And so basically you end up with 3 radioactive daughters, which can then redistribute throughout the body. And that also needs to be taken into account because this reallocation of daughters can induce additional side effects in terms of radiotoxicity. But how can we then evaluate the safety and efficacy? For that, we can, of course, do dosimetry. And so for dosimetry, we can basically use the concept of a source and target organ. Where basically if we're using alpha particles, the target organ is mainly limited to the source organ. And then we need to take into account these so-called S values, which are basically quantifying the mean absorbed dose to the target organ for each radioactive decay in the source organ. But once we have these, and we can actually, so when these can be calculated using Monte Carlo simulations, where we assume a simple geometry or even a voxel-based model. But once that we have these S values, we can even scale them based on the organ mass of the patients, such that we can do more patient-specific dosimetry. But so once we have these S values, we can do the dosimetry, but we still need to have actually the time integrated activity. And there, basically, we need to do a temporal sampling of the uptake in tissue or in organs, where we actually need to also do this on using an appropriate number of time points. And that is actually where imaging can play a crucial role. And this is shown here, where basically after the administration of the radiotherapeutic, you can actually scan the patients at several time points using a SPECT scanner, for instance, and where basically you get a good idea about the biodistribution of your radioligand. And then once you can extract the uptake in the different organs, and based on these time activity curves, so to speak, you can actually also estimate the time integrated activity. And so this is actually, this can then be used to actually do a dosimetry. But now focusing on actinium, why is actinium so challenging? Actinium has a rather complicated decay chain. So it has a long half-life of 10 days. And it actually generates 4 alpha particles. They are here shown in red. But during the decay chain, we only have a limited number of gamma emissions. Only the daughters of francium and bismuth are actually generating gammas. Francium is generating a gamma with an energy of around 220 keV, which is quite nice in terms of energy. But on the other hand, the branching ratio is very low. Bismuth has a higher branching ratio of around 25%, but on the other hand, we have to deal here with a high energy gamma, which is not that easy to handle actually. So, and so based on these 2 gammas, on the emission of gammas by the daughters, we can actually also quantify the concentration of actinium. But of course, there we need to make sure that we have a secular equilibrium between the mother and the daughter, which is perfectly feasible because the half-life of the mother is much longer than for the daughter. So at some point, we will reach a situation where the activity of the mother is equal to the activity of the daughter. For actinium, and specifically for francium, that can be achieved quite fast because the half-life of francium is only 5 minutes. So that basically means that after 20 minutes, so to speak, you are already at a level of 95% of the activity of actinium. If you need to start from a full ingrowth of francium. If you want to use bismuth, you have to wait a bit longer. You reach an activity level of 95% of the activity of the mother only after 3.4 hours. So that's is something that you need to take into account. But do we really need the gammas? Well, I just included this because I want to mention this also. Perhaps sometimes it's very useful for preclinical purposes. You can actually also use an alpha camera, which is very similar to autoradiography. And this was first introduced by Beck and Jacobsen in 2010, where basically they put the cryosections on a scintillating layer, and this was then coated on a polyester sheet. And so basically the cryosections were emitting the alphas. These were actually interacting with the scintillation layer, and then they were producing light. And basically then the light can be picked up with a system which is very similar to a microscope with a CCD. And so basically then you can also actually visualize the uptake of actinium labeled or compounds, or more in general with compounds labeled with an alpha emitter. And this is an example with astatine 2-1 labeled to trastuzumab, where they looked at the uptake in the kidneys. And where then if you sacrifice animals at different time points after administration, you can also have an idea about the changes of renal uptake over time. So that is something you could use. Of course, this can not be translated to humans, and that don't allow any in vivo measurements. Well, one of the things that we use frequently is the gamma counter for gamma counter measurements, which is basically still at our site also the standard approach for doing biodistribution studies. We have done now some efforts to optimize this actually also for actinium, where we use both the francium and the bismuth signal to actually quantify the actinium concentrations. And there was one group who also used Cherenkov light because of the interaction of the high energy photons with the tissue. So it generates some light. This has also been used in the past for PET tracers. And the advantage here is that you don't need a very expensive SPECT system or PET system, but you can actually just use a system which is also used for bioluminescence measurements. And so these systems are frequently available in labs. So that is one thing that you could consider. And so here they also use it to validate the pre-targeting strategy. But basically here you have the negative tumor, here you have the positive tumor, and then you see over time the increase in uptake in the tumor expressing the target. But of course, all these techniques have very limited translational potential. From that point of view, microspect is much more interesting on the preclinical level. And actually, microspect imaging allows you to do biodistribution studies in vivo. And I just show you this example to illustrate this. So there's actually a compound label with indium 111. And so basically then the animal can be imaged at different time points after administration. And then you can easily pick up the changes in biodistribution of the ligands, where again you see an accumulation in the tumor at later time points, while you see a clearance from the blood pool. So that is a very convenient method to also do biodistribution studies. So why hasn't that been considered for actinium-225? Well, there was one study actually who looked at this more closely. It was the study by Robertson, was published in 2017. And they actually looked at the vector system. So that is a microspect system from MyLabs. And that's not a big surprise because MyLabs is also using microspect to image a PET ligand. So they actually use collimated PET. And for this evaluation, they actually used two types of collimators. They used an ultra-high sensitivity collimator, and they also used an ultra-high resolution collimator, while the crystal thickness was still 3/8 inch. And so here you can see the energy spectrum that they could acquire with this system. So this is here the energy window from bismuth. This is the energy window for francium. And you see actually with the ultra high resolution collimator that you will get a lower sensitivity. So the count rate was lower, significantly lower, compared to the ultra high sensitivity collimator, which was already an indication that the design of the collimator is crucial. So they performed a series of experiments looking at different measurements of image quality. So they looked at the contrast, the noise, uniformity, also the resolution. Just to point out at this stage already that the activities that they used were quite high, even for the phantom measurements, especially if you're looking at a preclinical scenario. I think the activities, injected activities, are at maximum 1 megabecquerel. And also this is a system which has a very small field of view. So you have to constantly translate your bed in order to visualize your full phantom of your full animal. And so basically they had to perform 36 translations to image the phantom. And then for each of these shifts, they also acquired a scan for 40 minutes. So the total acquisition time was 36 multiplied by 40. So that basically translates into an acquisition time which is not feasible in an in vivo setting. So I'm not quite sure how representative these phantom measurements are actually for in vivo experiments. But anyway, they these are the data that they presented. So this is for the ultra high resolution colorimeter. And so, yeah, so the color bar is also a bit confusing. So the hot region is also in red color. The cold region is also in red color. But so the background here is cold, and these are the hot rods. And so you can see that you dramatically lose resolution if you use the ultra high sensitivity colorimeter at the expense of increasing the sensitivity. They also looked at the image quality using this specific phantom. So they actually have here a phantom with two cavities. So they have actually a hot cavity, and then they also have a cold cavity, which they basically just fill with cold water. And so then they looked at the profiles through these cavities to look at the recovery, and they also looked at the background. So they also looked at the noise in the background. And so these were all the results from this phantom experiment. And then you can see actually that indeed we lose some contrast recovery if we are switching from the high energy to the high sensitivity colorimeter. So this is mentioned here. So you get a drop in recovery, especially between the hot and the warm region. The drop in recovery or the gain in recovery was not that clear from the if we were comparing the warm to the cold region. So basically comparing these two regions. And that could be due actually to the reconstruction method, because iterative reconstruction sometimes struggles to reach convergence if you have a cold region surrounded by a hot background. So that could be an explanation here. And then also here, there was not really a significant improvement in the noise or in the background variability if you were using an ultra high sensitivity colorimeter versus an ultra high resolution colorimeter. But we must state also here that the ultra high sensitivity colorimeter was optimized for low energy photons, and the ultra high resolution colorimeter was more like a general purpose colorimeter, which was also usable for high energy photons. So the data here were also quite mixed. So it was not really conclusive whether the system could actually be used for in vivo experiments. We have done some experiments meanwhile at SEK, which has a similar system, but with a larger crystal. So they had a 6/8-inch crystal, which should increase the sensitivity as such. They also have a dedicated colorimeter for bismuth, but also there the data are not still very conclusive. Then they also looked actually whether they could discriminate between the bismuth signal and the actinium signal, which could be of relevance if you're looking at the recalled daughters. And basically what they did is they just measured a bismuth generator. So here you have the bismuth and the actinium, and then the bismuth got eluted. So then here in this compartment, bismuth was present. And here in this compartment, there was actinium present, but not in equilibrium with bismuth. And then they measured using both the francium window and the bismuth window. And then you can see that actually the signal using the francium window was quite constant, which is quite nice because your activity of actinium stays constant. So basically with the francium window, you can measure the actinium signal without being confounded by the bismuth signal. And then if they looked at the bismuth window, they can actually nicely see the ingrowth of bismuth in the lower compartments where you had this shortage of bismuth because of the elution. And in the upper compartment, you could actually see the decay of bismuth because actually here was the bismuth that was elevated from the generator. So this was actually a proof of concept that by using both the francium energy window and the bismuth energy window, you could actually discriminate between bismuth and actinium. From a clinical perspective, there are not so many data available in literature on actinium SPECT. There were some case studies being presented. These two case studies were from a patient who was treated with actinium PSMA. And so they acquired some planar data here, where they also looked at which energy windows they should use for the imaging. So basically, they considered three energy windows. They considered the energy window of bismuth, the energy window of francium, but also an energy window centered over the lead peak of the collimator. And so basically, they stated that the more energy windows, the more counts and the higher the image quality. But even then, I think these data are not really convincing that that could be feasible in clinics. There was another case study which seemed the most promising, and that was actually a case study by Gossowicz et al., where they where actinium PSMA was used as treatment after lutetium PSMA. And basically, they also did SPECT scanning of actinium PSMA. They used some of the kinetic data that they acquired with lutetium SPECT. So they acquired lutetium SPECT scans at different time points during the lutetium therapy. And they used this kinetic information to also estimate the 3D dose maps for actinium PSMA. And so these data look that they could be used in clinics. So that seems quite promising, so to speak. There was then finally another study who also did extensive phantom measurements and also evaluated different SPECT systems for actinium SPECT. And basically there they concluded that you should use a high energy collimator. So it was also with the Siemens system that the high energy collimator should be the collimator of preference. They also again looked at different energy windows, where they also stated that if you want to boost your sensitivity, you should take into account all the energy windows. But on the other hand, if you look at the signal to noise ratio, you should be more careful, because it's not only about acquiring as much counts as possible, you should also acquire counts that can be used. And so here, in terms of signal to noise ratio, actually you get the highest values if you use an energy window centered either over francium, and also the energy window centered over bismuth was not doing that bad. Unfortunately, they didn't look at scatter for this study, which is also quite important. And they also didn't look at the combination between the francium window and the bismuth window. So to have a kind of a roundup slide, so for actinium SPECT, so the challenge are there, the small injected activities, if you're using actinium-based treatments. So we are only looking at around 10 megabecquerel for patient treatments and lower than 100 kilobecquerel for preclinical treatments. The lack or the very low abundance of gamma emissions. So only the francium daughter and the bismuth daughter are generating gammas, but with a very low abundance. And then we also have the technical limitations, and then basically the sensitivity of the detector, because we are using a SPECT system where basically the sensitivity is reduced by the collimator. And so that's why we wanted to take advantage of the possibility of PRISMAP to actually try to maximize the potential of actinium SPECT by focusing on our clinical system. So we have an in-house Siemens Intevo system, which is a state-of-the-art SPECT CT system. And we would actually like to anticipate some of the upcoming clinical trials that will be performed at our center, and just make sure that we have the most optimal settings for actinium SPECT, so that once that we start to scan patients, we can actually get the most out of the data. And then we also have a microSPECT system at our preclinical facilities. It's actually a molecules system, where we also want to see what the potential is for actinium SPECT. So more specifically, for the Siemens Intevo system, we will consider two types of collimators also here, like a medium energy and a high energy collimator. The crystal thickness is again 3/8 inch, where we, in comparison to the previous study where we will look closely at scatter, where we also consider spectral reconstruction, which basically means that we will take into account the different energy windows during the reconstruction. So we will not reconstruct each of the energy windows separately, but we will do one reconstruction while taking advantage of the different energy windows. And where we also, because we are taking into account the different energy windows, where we will also try to discriminate the signal from actinium by using the Francium window from the signal of free bismuth. And that can be quite important, again, because of the recall daughter effect. And this is actually a study that shows this. So basically, they did a biodistribution study shortly after administering the radiotherapeutic. And then they did again a biodistribution after 4 days. And they actually could see a significant increase in the kidneys. And they explained this actually because of the recall daughter effect and because of the free bismuth being redistributed and being taken up by the kidneys. So I think for patient study, it would be quite nice if we could also do this monitoring in vivo. And then for the molecules SPECT CT system, we are currently discussing with molecules to actually refurbish this system, because now it's optimized for low energy gammas, to actually refurbish this system for high energy gammas, where we will probably not only need to look at a different collimator, but also probably perhaps use a thicker crystal, and perhaps also increase the shielding. And that once that this refurbishment has been done, the radioisotope that will be delivered by PRISMAP will actually be used to actually evaluate the system performance, such that this system can also support future developments of new therapeutics who are using actinium. So, and this was actually the end of my presentation. I saw that in the other presentations, they also shortly mentioned how they got in contact with PRISMAP. For our case, it was actually because I'm part of KU Leuven, but I was not part of PRISMAP. So I actually got to know this consortium from my colleagues here at KU Leuven. And since I'm more involved in the research things, I think it was a great opportunity to have access to radioisotopes, which are otherwise difficult to get your hands on, and especially with actinium, and especially since we have a research track here on actinium, it's a nice opportunity to have access to actinium. Okay.
Okay, so thanks a lot for this testimony. Do we have some questions in the audience or online? I have a small remark. You said in the beginning that actinium itself does not emit any gamma rays. Obviously, it does some weak gamma rays, which are not useful for imaging, but for germanium spectroscopy to know exactly how much you ship out.
Yeah, you're completely right.
So, any more? And I invite the people who are still thinking about their questions to afterwards use the chat. You can also directly contact the speakers or just continue discussion while we go on with our session. So I will return to the microphone to Thomas now. Thanks a lot.
Okay, thank you very much. Thank you to our three speakers today for their testimonies. It was very enlightening to see the variety of topics that they cover themselves, and also the process they went through in preparing their proposals and how they came across PRISMAP. I think it's very inspiring for all of us. And thank you also for Uli to put that session together. So now we come back to what I guess a lot of you are really expecting and waiting for, and that is what